Okay.
Now I talk about the topic cell division. What is cell? Cell comes from pre-existing cell. Cell. Therefore, cells need to make more cells are called cell division. The divisions of cells into daughter cells is called cell division. The growth and development of every living organism depend on cell division. The new cells are called daughter cells. Daughter cells can grow and increase in size until they divide and produce two more daughter cells. This process continues, resulting in organism growth and reproduction. Next, what is cell cycle? The cell cycle is a series of events that take place in cell leading to duplication of its DNA, that is DNA replication, and divisions of cytoplasm and organelles to produce two daughter cells. It is a growth and divisions of a single cell into daughter cells called replication, that is duplication. In prokaryotic cell, the cell cycle occurs in the process termed binary fission. In eukaryotic cell, the cell can be divided into two periods. One is interphase and mitosis. First you have to see about interphase. In interphase they are divided into three sub phases. One is G1, S and G2. Next G1. G1 it represents as a pre-synthetic phases. S is represent as a DNA synthesis. G2 as a pre-mitotic phases. First one, interphases. During these phases, the cell grows accumulating nutrients needed for mitosis and duplication its DNA. Next, mitosis or M phases, during which the cell split itself into two distinct cells. The durations of the cell cycle varies from hours to years. For example, human cell has a durations of 90 hours. The phases of cell cycle, that is G1, S, G2 and M phases. Cell cycle consists of three phases. G1, that is growth and synthesis and primary growth phases. S phase, DNA synthetic phase and DNA is replicated. G2, preparations of division, it is a secondary growth phases. M phases including the overlapping process of mitosis and cytokinesis. Next we are see about G1 phases. G1 phases begins at the completions of mitosis and cytokinesis and lasts until the beginning of S phases. In this S phases, replications of chromosomes is restricted to one specific portions of interphases called S phases, that is DNA synthetic phases, which is typically last about 6 hours. Next phase you are going to see G2 phases. In the G2 phases, portions of interphases that follows S phases is called gap phases 2. Next we are see about M phases. The M phases include the overlapping process of mitosis and cytokinesis. Next we are going to see about mitosis. Mitosis divided into the 5 stages: prophases, metaphases, anaphases, telophases and cytokinesis. First you are going to see about prophases. In the prophases, the chromosomes are coiled, the nuclear membrane are disintegrated, spindle fibers are formed. Next we are going to see about metaphases. In that metaphases, the nuclear membrane is completely gone, the chromosome become aligned. Next you are going to see about anaphases. In that anaphases, the chromatids are separated, the number of chromosomes are doubled in number, then they are pushed and pulled toward opposite pole by the spindle fibers. Next we are see about the telophases. In the telophases, the cells are divided into two, the chromosomes are uncoiled, nucleus are reformed. The spindle formation and the apparatus are disassembled in the stages. Next we are going to see cytokinesis. This is the last stage. In this cytokinesis, this is a process that the cytoplasms of a parent cell is divided between two daughter cells produced either in the via of mitosis or meiosis. Thank you.